All right, last couple of days in class we've been talking about photosynthesis, and so I thought I'd make a little short video to go over what we've discussed. It's a pretty complicated process that hopefully if you watched the video over cellular respiration, there'll be some terms and some that are kind of familiar, and it makes it a little bit easier to understand, but there are some new concepts to learn about in photosynthesis as well. First of all, just remember when we talk about photosynthesis, remember that it occurs in plants, algae, and bacteria. Those are autotrophs. They take basically the products of cellular respiration, water and CO2, and convert that into glucose, other organic compounds, and they give off oxygen in the process that we as heterotrophs take in to do cellular respiration. And so that continues that whole cycle that we've already discussed with cellular respiration. Here's the balanced chemical equation for photosynthesis. Remember, it's just the opposite of cellular respiration. So we have 6 carbon dioxide molecules are going to combine with 6 water molecules in the presence of sunlight to form glucose, which is stored energy, and oxygen. And remember that stored energy and glucose is going to be released through the process of cellular respiration, and that's how those two processes go hand in hand. The major light-absorbing pigment that are found in chloroplasts are chlorophyll, and those molecules, what they do is capture that sunlight energy and convert that to energy, usable energy for the plants. The primary pigments that are found are chlorophyll A and chlorophyll B. What I wanted to point out from this little image here is the wavelengths of light that they absorb the most light from. So you'll notice chlorophyll A and B really absorb a lot of light in that blue, indigo, violet range of the visible light spectrum. They also absorb a little bit in that red, orange, and yellow spectrum. But you'll notice here in the green, they don't really absorb any of that light because they are reflecting that light. And that's why chlorophyll is green, because it reflects the green light. There's another pigment called the carotenoid here, and what they do is kind of extend that visible light. And they will absorb some of that green light that the chlorophyll A and B cannot. But if you'll notice, once you get out here past the green and to the yellow, the oranges and reds, they're absorbing, sorry, I said that wrong. They are reflecting those lights. Now, we typically don't see carotenoids until late in the year during the fall when plants stop photosynthesizing, they stop producing chlorophyll A and B. And these pigments will start to show up, and we see that in leaves of plants, and they turn yellow and orange and red in the fall before they actually completely go dormant and completely drop off of the leaf, off the tree. But those are those pigments. Now, there's an experiment that we talked about in class. It's really cool. It's from a scientist by the name of Engelmann, and he conducted this back in the 1800s before any technology that would tell scientists show scientists how plants absorb different colors of light. He had some filamentous algae, and he placed it under a light that passed through a prism. He also took some bacteria that was aerobic bacteria, and aerobic, if you remember from cellular respiration, means that oxygen is present. So this aerobic bacteria really like oxygen. So he passed this light through a prism, and he shone that onto this algae. What he noticed was the algae would accumulate more in that blue, indigo, violet, and in the red and the oranges over here, and yellow a little bit. Not a lot in that green. And what that showed was that those were areas that where a lot of photosynthesis was taking place. And so the light was being absorbed in photosynthesis, and he knew that because the bacteria would be attracted to that oxygen that the plant was giving off through photosynthesis. And so the bacteria is accumulated in abundance here and in here. Not so much here because, again, that color was being reflected by the algae and not absorbed. So it's a pretty cool experiment that he did way back a long time ago. All right, so if we look at the light reactions, and there's two parts to this photosynthesis. There's a light reaction and the Calvin cycle. The light reaction, just in general, takes sunlight and water and converts that sunlight into ATP and NADPH. We'll talk more about that in a second. They release oxygen in the process. They actually split that water molecule. And they provide the energy through ATP and the electrons and NADPH to power the Calvin cycle, which basically takes CO2 and converts it to glucose. And that's kind of it, the big picture of what we're talking about. There's a video. It's by Ricochet Science, that if you want to check that out. I'm not going to play it here just for time purposes, but you can go in and you could look at Amoeba Sisters or this video on photosynthesis, the light reaction, to help if you're confused with it, and maybe help you explain it to you in a little more detail. It's not an easy process, so hopefully this video will help you a little bit. Now we're looking here at the leaf. The leaf is the primary organ of photosynthesis for plants. They have that surface area that allows them to absorb all that sunlight. On the bottom side of the leaf you have these stomata, and stomata are little openings that will open and allow carbon dioxide to enter and oxygen to be released. And this is a look at an actual chloroplast, and you'll see the chloroplast is made up of thylakoid, and we'll talk about the thylakoid membrane in a little bit, which will look kind of like little poker chips. And then you have stacks of thylakoid called granum, and then there's this fluid-filled area space around that called the stroma. And so those are some terms that are going to be referred in these two processes. All right, if we look at, I want to come back to this in just a second. If we start off here, this is the light reaction. Now you'll notice there are two photosystems, photosystem 1 and photosystem 2. Photosystem 1 was named first because it was discovered first, but it actually occurs second in the process. Photosystem 2 actually occurs first. What happens in photosystem 2, and this is a reaction protein here where all this is taking place, has these little green buttons are chlorophyll molecules. So what happens here, light will strike the chlorophyll, and right here there will be some electrons that get excited by the light. The photons excite those electrons, and they kind of excite them to this primary electron acceptor here. So when they pass from this P680 chlorophyll to this primary electron acceptor, a water molecule is going to be split in order to replace those electrons. And during the splitting of that water molecule, that's kind of what we're talking about here. Plants use enzymes to split a water molecule in order to utilize electrons for the light reaction. And then oxygen is going to be released as an unneeded byproduct of that splitting of the water. So once those electrons get excited and they go up to this top of this primary electron acceptor, they're going to go down an electron transport chain. This electron transport chain is then going to generate ATP, very similar to the electron transport chain that was found in the matrix, the inner membrane, the membranes of the matrix of the mitochondria. Couldn't spit it out there. And which generates ATP. The same process is basically going on there, chemiosmosis, and we've already discussed that. So the main thing we want to know is that ATP is generated through that electron transport chain. Then those electrons are going to pass to photosystem 1, and again, light is going to strike the chlorophyll, excite those electrons to another primary acceptor here, and then they get passed down a second, shorter electron transport chain, and those will get transferred to NADP+, which is very similar to NAD+, that we saw in cellular respiration. And then those electrons get passed there, forming NADPH. And that, again, is this molecule that's going to carry electrons. And they carry those to the next step, which is going to be the Calvin cycle. So the Calvin cycle energy from the light reaction is used along with CO2 to make glucose and other organic molecules. And it kind of looks like this. It's a pretty complex process. We're not going to spend a whole lot of time and a lot of detail, but what I want you to understand from it is we're going to start with this RUBP, which stands for ribulose biphosphate. It is a 5-carbon compound. And what happens here is carbon dioxide is going to get added to that, forming this 6-carbon sugar. This is what we call carbon fixation. There is an enzyme called rubisco that adds that carbon to that ribulose, that RUBP. Rubisco, by the way, is the most abundant protein found in chloroplast, and therefore a lot of people can probably think it's the most abundant protein found on Earth. So that's pretty cool. This gets split into 2 3-carbon molecules, and then this process of reduction. Remember, reduction is the gain of electrons. Electrons are going to get passed to that 3-carbon molecule, and ATP is going to be used to form a what's called G3P, which is a 3-carbon molecule. It's going to take 2 G3Ps to form a glucose, which is a 6-carbon molecule. Okay. Then the remaining. Now, one thing I wanted to mention here. This is showing you actually 3 CO2s at a time. So I like to kind of count the carbons. If you'll notice in this RUBP, we have a 5-carbon molecule. And so if you have 3 of them, that's 3 times 5 is 15 carbons. And then we add 3 CO2s, that's going to be 18 carbons. That's going to be changed to 3 of these 6-carbon molecule, intermediate molecules. So there's your 18 carbons. That's going to split into 6 of these 3 PGAs. I know it's confusing. And then of those 6, only 1 is going to be leave the whole Calvin cycle. And the other 5 are going to be regenerated. They're used to regenerate the 3 RUBP. I know it's kind of confusing, but what I want you to really know is that it takes 1 complete cycle to just add 1 carbon to that glucose. So in order to form a glucose molecule, you actually have to have 6 turns of that Calvin cycle, 1 CO2 added at a time. And it should make sense because glucose is a 6-carbon molecule. So it's going to take 6 CO2s to make that 1 glucose. And you see that in that chemical equation. Real quick, I'm running low on time. I don't want to run out of time before I go over this. This is kind of summarizing these 2 processes. Remember, the light reaction is going to occur in the thylakoid membranes of the chloroplast, in which light energy is going to be converted to ATP and NADPH, and water molecules are split to provide the electrons for this whole process, and oxygen is released. The ATP and NADPH generated by the light reaction is going to enter the Calvin cycle and power the production of glucose. Now, CO2 is going to be added to RUBP, forming this G3P molecule. And again, a couple of those G3Ps will form a glucose. A couple of other things here. Remember, it takes place in the stroma of the chloroplast. It uses ATP and NADPH to convert CO2 to G3P. We've discussed that. Then the ADP, because after the ATP loses a phosphate, it becomes ADP. And then the NADPH is going to lose an electron and become NADP+. These 2 are going to be sent back over to the light reaction to pick up an electron and to pick up another phosphate group and become ATP. So that is photosynthesis real quick. Okay, so I've gone over it real quickly. I would encourage you to go online and watch some videos if you're still confused with it. I'm going to be uploading a worksheet tomorrow, hopefully to help you understand it. Again, I can't encourage you enough to do these worksheets yourself. Don't look up answer keys online. I know that's a temptation to do, but hope you won't learn much from that. And feel free to email me if you have any questions on this process of photosynthesis, because it can be a confusing process. So I hope this helps a little bit, and again, let me know if you have any questions.